Hi, I'm Jennifer, an MCAT teacher and content creator at Kaplan. In this video, Nick, one of our expert instructors, will take you through a passage that we've identified as one of the toughest passages on Kaplan's free MCAT practice test. This test was designed by Kaplan's question writers and validated by Kaplan's psychometrics team to be as representative of the actual MCAT as possible, so you know you're getting a great representation of what you will see on test day. This passage from the Bio-Biochem section focuses on some biochemical experiments investigating a protein. Follow along to see how Nick attacks this passage using Kaplan's passage and question strategy.
Diving into this passage, I see two figures with graphs. This tells me this is going to be an experimental passage, so I'm going to interrogate, really taking the time to understand what's going on in the experiment. With dense experimental passages like this one, I like to write notes down to help keep myself organized. Starting with the first paragraph, I'm introduced to MT Mycobacterium tuberculosis and its gene TLYA, also called RV1694. I'm already focusing on making clear in my mind what these abbreviations are for, since I know from my preview that they'll come up throughout the rest of the passage. Importantly, I see that the last sentence sets the scene for what is to come experimentally, characterizing the MT gene. The second paragraph discusses a little more on the TLYA protein, which the first paragraph also labeled as RV1694. There are a lot of fluffy details here. I'll read through them all, but I'm not going to stop and get bogged down trying to understand everything about them either. Instead, I'll make a note of the main idea, that TLYA has two enzymatic roles as an rRNA methyltransferase and a red blood cell destroyer. In the next paragraph, a keyword immediately catches my attention: hypothesized. I slow down to read it carefully and note the goal of the experiment is to determine if two cysteine residues are important for TLYA's hemolytic activity. The passage describes that in this experiment, blood cells were incubated with TLYA in two conditions, native and reducing conditions. I remember from my studying that reducing conditions break disulfide bonds between two cysteines, which are the amino acids the researchers are investigating. The readout for enzymatic activity is absorbance. More lysis means more hemoglobin release, and more hemoglobin release means more lysis. When I get to the graph, I read the figures legend and axes first. The y-axis is absorbance, how much hemolysis there is, while the x-axis is TLYA protein concentration going from higher to lower levels. I see that the graph for native conditions is higher than the reducing condition, meaning reducing conditions decrease hemolysis. Thus, it supports the hypothesis that cysteine residues may be important for hemolytic activity. A second experiment described in the following paragraph looks at how TLYA affects translation efficiency. This experiment takes ribosomes from E. coli that were either given TLYA or a mock vector as a control, adds components for translation, and then measures the percent translation with or without capreomycin added. The graph looks complicated, but I'm going to take the time to break it down. I read the figure legend first, which can sometimes simplify what is said in the passage. The y-axis measures translation percent, but this is where detailed reading is very important. I see that the translation percent axis is flipped, with 100 at the bottom rather than the top. Next, I see the x-axis is time. Finally, I take a glance at the legend to figure out what the specific conditions of each line is. Now I'm ready to look at the graph results. First, I start with mock vector and TLYA without capreomycin. Both have similarly high translation over time. Next, I look at mock versus TLYA at a 16 nanogram per milliliter with capreomycin. The translation isn't much lower with the mock vector, but it is much lower with TLYA. Finally, mock vector and TLYA at 80 nanograms per milliliter with capreomycin have the lowest rates of translation, with TLYA having the lowest translation efficiency. Overall, these results suggest that TLYA leads to decreased translation in the presence of capreomycin. These are the notes I've taken on the passage, short but to the point, and telling me in quick form what the figures show. Taking these notes helped me focus on the important details while reading, and will now help me answer the questions without having to redo analysis. The first thing I noticed after clicking to question 51 is the varied answer choices. Reducing, enzymatic activity, there's no apparent pattern. Looking at the question stem, it asks for a detail from the passage. TLYA binding to red blood cells does what? Having interrogated the passage, I recall that hemolytic activity of TLYA came up around the second paragraph of the passage in Figure 1. Going back to paragraph 2, I skim until I find where the hemolytic activity is mentioned. The paragraph makes note that TLYA is a monomer when free in solution, but becomes an oligomer when bound to cell membranes. Going back to my notes on Figure 1, which was looking at hemolysis in reducing and native conditions, I noted down that reducing conditions decreases hemolysis. With the information from paragraph 2 and Figure 1, I'll go to the answer choices and begin to eliminate what doesn't match. Starting with answer choice A, there's no evidence for this. The passage never actually mentions what affects TLYA binding to membranes, though Figure 1 does show that reducing environments decreases hemolytic activity. This is not my answer. Answer choice B looks nice at first until I get to the last word, monomers. Paragraph 2 says TLYA oligomerizes when bound to the cell membranes rather than staying as a monomer, so this statement is unsupported by the passage. This answer can go in the trash. C is a direct opposite from what is stated in paragraph 2. Another wrong answer, which leaves me with answer choice D. During the test, I'm going to pick D and move on without any more thought, but in review, I can look at why it's correct. Paragraph 2 states TLYA oligomerizes when bound to cell membranes. Figure 1 showed reducing conditions which break disulfide bonds, leading to a reduction in hemolytic activity. Collectively, this suggests oligomerization occurs via disulfide bonds, and when oligomerization is disrupted, so is the hemolytic activity of the protein. The answer to question 52 immediately stands out with different biological molecules listed. Ones I recognize, so I'm ready to do this question, especially given the short question stem. The question is a detail question asking what would be included in the buffer in experiment 2. Going back to the paragraph on experiment 2, I scan for where a buffer was mentioned. The end of the paragraph states that the buffer contains necessary biological components needed for translation. I know translation is the process of turning mRNA into protein, so the buffer has to contain something used in translation. Since this is a broad question, I'm going to use elimination to find the correct answer. DNA is definitely not correct. DNA would be used in transcription and replication, not translation. GTP looks good. From my content knowledge, I remember that GTP is used in translation for energy in the elongation and translocation steps. This is my answer. But had I not been sure, I could check out C and D. C is wrong because NADH is not used as a source of energy for translation, but rather used in the ETC to create ATP. And helicase is used to unwind DNA, not in translation. rRNA methylation stands out in these answer choices. And going into the question stem, this looks to be a data interpretation question, since one of the figures from the passage discussed capreomycin. Specifically for this question, I need to answer the question of how TLYA rRNA methylation could affect translation in the presence of capreomycin. Going back to my notes, I wrote that TLYA decreases translation efficiency in the presence of capreomycin. This seems like a sufficient summary to start answering the question without looking back at the graph. Thanks, past me. Because in experiment 2, more TLYA, which the passage states is an rRNA methyltransferase, leads to decreased translation in the presence of the antibiotic capreomycin, my prediction will be rRNA methylation decreases translation. Looking through the answer choices, there isn't anything that directly matches. However, A, I know is incorrect because rRNA methylation is involved, and B is opposite of what I predicted. Looking more closely at answer choice C, I see that it's the flip of my prediction. I predicted rRNA methylation decreases translation. So it makes sense that a loss of rRNA methylation in these cells could reasonably be expected to increase translation efficiency. Answer choice C is therefore correct. The short answer choices first stand out to me in question 54, as well as the units of kilodalton, meaning this question will be a detail on something, probably TLYA's molecular weight. The question specifically asks about the possible molecular weight of TLYA when bound to target cells. I remember from the passage it stated TLYA binds to red blood cells specifically. So I can rephrase this question as, what is a possible molecular weight of TLYA on red blood cells? My notes don't have anything on molecular weights, but I do remember when going through the passage that paragraph 2 specifically had information on the TLYA protein. Going back to paragraph 2, immediately I see that TLYA is a 28 kilodalton protein. Reading on to where the paragraph discusses its effects on red blood cells, I also read that TLYA goes from a monomeric form to an oligomer when bound to target cell membranes, meaning multiple 28 kilodalton monomers are combining to create a larger protein. Going back to the question, I also take note of the word possible molecular weight. The passage didn't say specifically how big the oligomer is, so that means I can predict that the answer will be a multiple of 28. Going to the answer choices, A is technically a multiple of 28, but this is the monomer form, not it. 60 kilodaltons is not a multiple of 28, rather 56 is. B is also not it. C is a multiple of 28. I found my answer and don't need to look at D, though a quick glance tells me that it doesn't fit. Question 55 immediately stands out as a data analysis question, given figure 1 is mentioned in the question stem. This question is more specifically asking what would happen if two cysteines turned into alanines. I like to think of these as extended data analysis type questions, as this question is not only asking for an understanding of the results of figure 1, but also to apply these findings to a related but new situation. Heading to my notes on figure 1, I see that I wrote reducing conditions decrease hemolysis. When I was going through this figure, I thought about how reducing conditions break disulfide bonds. Similarly, changing cysteines to alanines would lead to a lack of disulfide bonds, which I would predict would mean a decrease in hemolysis. This prediction perfectly matches choice B. When I click to question 56, I note that the answer choices are all parts of a cell. The question stem provides a new scenario not in the passage where TLYA is tagged with a fluorescent marker and put into cells. The question that follows is testing details from the passage about TLYA, asking where TLYA would not be expected to be found in a cell. And this language is something I would make a note of. I need to remember that this is an except question to avoid making a silly mistake. This question requires knowledge of the function of TLYA in order to predict where TLYA would or would not be found in a cell. I recall from the passage that TLYA both acts as an rRNA methyltransferase and a hemolysin. rRNA, or ribosomal RNA, is found in ribosomes, which are in the cytosol of the cell. So TLYA would expect it to also be in the cytosol if it acts on rRNA. As a hemolysin, TLYA pokes holes into the membranes of red blood cells. This means TLYA is found outside of cells, and therefore likely to go through the secretory pathway, also called the endomembrane system of the cell. In this pathway, proteins that are slated for secretion outside the cell are translated at the rough endoplasmic reticulum before moving through the Golgi and through exocytosis. At this point, I know that TLYA would be expected to be found at least in the cytosol, the rough ER, and the Golgi apparatus. Because this is an except question, when I go to the answer choices, the only part of the cell that is not on my predicted list of places for TLYA is the nucleus. This is my except answer, meaning A is the correct answer.